Okay, so let's use this mind map on glycolysis to try and brainstorm the pathway and its implications and applications too. So there is a metabolic strategy, and central to energy metabolism is the maintenance of a blood glucose concentration of about 5 millimoles, which is essential for normal cerebral function. So confusion and coma can result if blood glucose concentration falls below 3 millimoles, while serious vascular damage may occur if it exceeds 8 millimoles for significant periods. So after a meal, the glucose concentration in the portal venous blood can easily reach about 20 millimoles, and much of this excess would be removed by the liver. Stimulation of insulin release results in the uptake of glucose by the peripheral tissues, that's the muscles and adipose tissues. And surplus glucose is stored locally in tissues as glycogen, but mostly it's converted into fats. And this level of glucose, that's the 5 millimoles, is actively defended by the liver, which removes glucose when too high and replenishes it when too low. So both the supply and demand for glucose may vary more than 20-fold over 24-hour period, but can change suddenly and sometimes without warning. The liver can both um, uptake and also secrete the glucose, and it is one of the few tissues in the body to permit bidirectional glucose transport, as in enterocytes and kidneys. Also, um, others I can think of, the enterocytes in the intestinal absorptive cells. So with the overall response of to blood glucose levels, realize that when it is too high, um, as with the physiological values of parameters more than 6 millimoles, there's a conversion to glycogen. That's when glycogenesis takes place. That's with a short-term response, but with a long-term response, they make triglycerides. That's the lipogenesis. But when it's normal, say 5 millimoles, um, we are dealing with the blood itself, not generally. So there's no action taken both short term and long term. But when it's too low, for example, there's a conversion of the glycogen to glucose, that's gluconeolysis. And also with the long term, that's when the gluconeogenesis takes place with the making of glucose. So the transport of glucose into the cells, realize that they cannot enter the cells by simple diffusion, they enter by facilitated diffusion and um, co-transport. So with the facilitated diffusion, transport through the membrane, um, in some tissues like that of the liver, erythrocytes, adipose tissues, brain, muscles, is carried by is by carrier mediated diffusion, is driven by the concentration gradient. With the facilitated diffusion, there are transporters like GLUT1 and GLUT14, variable tissue expressions too, and the transporters have two conformational states in membranes, like glucose binds, alters the conformation and releases glucose into the cells. In adipose tissue, the skeletal muscle glucose transport is enhanced by insulin, that is by increasing the number and activity of GLUT4 transporters. So the co-transport, the cells of the intestinal mucosa, renal tubules, and the choroid plexus, for example, transport glucose against gradient, and that's energy dependent. So the energy dependent process, that is driven by the sodium gradient across the membrane. The carrier is a sodium dependent transporter, or SGLT, and understand the there are several metabolic steps of the glycolytic pathway, and um, with the starting material and the end product of glycolysis, they all play a role in the maintenance of glucose levels too. The key regulatory steps of glycolysis, to try and use this mind map to remember them. And the effect of pyruvate in the presence and absence of oxygen, and also differences between the gross and the net products of ATP producing glycolysis. Metabolites are needed for the continuous operation of glycolysis. So glycolysis actually plays a key role in energy metabolism by providing significant portion of energy utilized by most organisms. So generally know from glucose to glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 base phosphate to 1,3 base phosphoglycerate, 3 phosphoglycerates to 2 phosphoglycerates, and then phospho enol pyruvate, and then finally to pyruvate. But we'll take our time, we'll go through the steps step by step. So the 10 steps of glycolysis and the enzymes catalyzing them, we just try and remember them. The first step being the glucose hexokinase is the enzyme involved here, converting it to glucose 6 phosphate. And then the enzyme here at the second step is the glucose 6 phosphate isomerase. And the third step is the fructose 6 phosphate, and you have your PFK1, and that's the first committed step. And the fourth step is the fructose 1,6 base phosphate. That's when aldolase comes in. So the first 4 steps, that's the hexose stage. You have 2 ATP consumed, and the location of the first 4 steps is the cytosol. Yeah, so the fifth step being the other steps also takes place in the cytosol. So always remember that with the glycolytic pathway, everything takes place in the cytosol. Alright, so let's go on with the fifth step that has to do with hydroxyacetone phosphate, and there you have your triose phosphates isomerase, and with 6, that is glyceraldehyde 3 phosphates, and that's the enzyme here is the glyceraldehyde 3 phosphate dehydrogenase. And the 7th step had to do with 1,3 base phosphoglycerate, and there you have your phosphoglycerate kinase. And the 8th step being 3 phosphoglycerate, and the enzyme there is the phosphoglycerate mutase. The 9th step being 2 phosphoglycerate, and you have your enolase, which removes the H2O, the water. And the 10th step being phosphoenol pyruvate, or the PEP, and the enzyme here is a pyruvate kinase. And the 11th step, or the last step, being pyruvate. So from the 5th step to the 11th step, you have your triose stage, and therefore ATP is being produced. So that is something very important to remember. Okay. And you could also note the phosphorylation level starting at step 7, 1,3 base phosphoglycerate. Alright, so with the general functions of the glycolytic pathway or glycolysis is to provide ATP and its energy, and also generate intermediates for other pathways like the hexose monophosphate pathway, glycogen synthesis, and can think of pyruvate dehydrogenase in fatty acid synthesis and the Krebs cycle, and also glycerol phosphates in TG synthesis. Alright, so with the glucose, you have your glycolysis with the 10 successive reactions. You get 2 pyruvates, and then it could go on to ethanol fermentation or lactic acid fermentation, with the ethanol fermentation being anaerobic. Under anaerobic conditions, you have 2 ethanol and 2 carbon dioxide being produced. That's fermentation to alcohol in yeast. But with the 2 pyruvates, still under anaerobic conditions, produces 2 lactic acid or lactates as the lactic acid fermentation. The fermentation to lactate in rigorous contracting muscle erythrocytes and other cells, and also in some microorganisms too. But remember, 2 pyruvates under aerobic conditions, 2 carbon dioxide comes out of the reaction form two acetyl CoA, and that leads to the citric acid cycle. 4 carbon dioxide is being produced, 4 H2O. Water is also being produced, and it normally happens in animals, plants, and many microbial cells under aerobic conditions. Right, so with the degradation of glucose via the glycolytic pathway, remember stage 1 has to do with the reactions from 1 to 5, steps 1 to 5, and the stage 2, the reactions from steps 6 to 10. Alright, so remember the preparatory phase where there is phosphorylation of glucose and its conversion to glyceraldehyde 3 phosphate. Yeah, so the first reaction where glucose is converted to glucose 6 phosphate, ATP is being used over here, and then once the phosphate group is being used, you get ADP, and there's a first priming reaction, and then the second being glucose 6 phosphate to fructose 6 phosphate. The second priming reaction happens over there, and the third where there's cleaving of the 6 carbon sugar phosphate to 2,3 carbon sugar phosphates. That's step 3, glyceraldehyde 3 phosphates. And with the payoff phases where there's oxidative conversion of glyceraldehyde 3 phosphate to pyruvate and coupled formation of ATP and NADH. Yeah, so also remember the non-redox reactions in the various phases, and the nucleophilic attack of the 6 carbon, and also the OH group of glucose on the gamma phosphates of the MJATP phase. That is when there is phosphorylation in step 1 by hexokinase, where magnesium is being utilized over here too. Alright, so the enzyme that transfers phosphate groups between ATP and metabolites are kinase, and by here too with an enzyme kinetics, you can think of induced fits in the hexokinase happening with the D-glucose, and there's a closing of 2 loops places in the ATP in close proximity to carbon 6 and also the OH group, and it excludes water from the active site. So the glucose induces a large conformational change in the hexokinase, and the substrate-induced conformational change is responsible for the enzyme specificity. And also with the conversion of glucose 6 phosphate to fructose 6 phosphate, magnesium is also utilized over here too. That's the enzyme, like I said, is phosphohexose isomerase. And then with the other steps, try and remember all the enzymes over there. So from the first step to 1,3-base phosphoglycerate, NADH is being produced, and then the ATPs to from there to 3-phosphoglycerate, 1 ATP, and from PEP to pyruvate to ATP is being produced. So you have about 4 NADH over here. That is with all the various reactions like the citric acid cycle, the lactate, and then when it comes to the production of ethanol too, you realize NADH comes into the reaction and produces NAD plus, NADH with from pyruvate to lactate, NAD plus too is being produced there. Okay, so just a quick one. I'll just use the acronyms or the short form of these two, like the PFK2, so that we can quickly remember the other upregulation and downregulatory processes like phosphofructokinase 2, um, PFK2, fructose 2,6 bisphosphate, F26BP, and phosphofructokinase 1. So these are three molecules that play key roles in glycolysis and its regulation, particularly in response to energy needs. So the PFK1, that's the fructokinase 1. It's a key rate limiting step or enzyme in glycolysis. It catalyzes the conversion of glucose 6 phosphate to fructose 1,6 bisphosphate using ATP. The regulation is activated by AMP, ADP, and F26BP, that's a fructose 2 6 bisphosphate, and is inhibited by ATP and citrate, indicating high energy levels. And with the phosphofructokinase 2, that's a PFK2, it's bifunctional enzyme with both kinase and phosphatase activities. It regulates the glycolysis by producing fructose 2,6 bisphosphate from fructose 6 phosphate, and with this regulation it's active in fed states. That's insulin activates the PFK2, leading to increased F26BP, which stimulates PFK1 and promotes glycolysis. So it's inactive in the fasting state, as glucagon phosphorylates and inhibits PFK2, reducing F26BP, thus slowing down glycolysis and favoring gluconeogenesis. So with fructose 2,6 bisphosphate, it's a potent allosteric inhibitor of PFK1, overriding ATP inhibition and ensuring glycolysis continues even when ATP levels are high. And with its control, it's being produced by the kinase domain of PFK2 is degraded by the phosphatase domain of PFK2, and with high F26BP, glycolysis increases, and with low F26BP, glycolysis slows down. Gluconeogenesis takes over. Alright, so that is something brief about the regulation. Okay, so the synthesis of the enzymes at three major steps is increase in response to insulin and decrease in response to glucagon. So glucokinase, PFK1, pyruvate, and think of them. So after a high or carbohydrate-rich meal, these enzymes are upregulated and glycolysis is favored. But in contrast, during fasting or in diabetes, high glucagon levels and low insulin levels will result in lower levels of these enzymes and less glycolysis. The insulin regulation of the glucose transport and diabetes, you realize that GLUT4 receptor is insulin sensitive and occurs in skeletal muscles and adipocytes. Other tissues have insulin independent glucose transporters and is terminated by receptor dephosphorylation. With type 2 diabetes associated with obesity, insulin resistance, which may be caused by secretion from adipose tissues like leptin and levels of free fatty acids, hyperglycemia with insulin production that is insufficient to control it. Finally, beta cells deteriorate, and with a possible treatment with increasing has to do with increasing expression of GLUT4 receptors, decreasing GLUT4 removal from the cell surface, and with the glucose transporters, they're recruited to the cell membrane by insulin signaling, and insulin binds to its receptor on the cell membrane. The insulin signaling causes glucose transporters to be recruited to the cell membrane. The glucose transporters transport increased amounts of glucose into the cell, and when the insulin levels are low, the glucose transporters are imported into the cell and stored. Alright, so that's an overview of glycolysis using the mind maps. They're more to read, but we can further on read about them.